Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.
Mastodons and mammoths. You'll see plenty of them in a few moments, and I'll try to clarify for those of you that may be a little fuzzy on which is which and exactly how they relate to one another in time and space. But I think you all know that they're big elephant-like animals. In fact, they're both relatives of elephants. And when you see the skull of one, this is the skull of a mastodon, it's quite clear that there are those big tusks that look much like the tusks of elephants. And although you don't see the soft tissues here, emerging from that hole in the forehead are the narial, in the live animal, the narial passages, and in fact the trunk that's very much like an elephant's trunk. Some of you may know that, that fossil mammoths and mastodons and so forth were what was found by early people, circum-Mediterranean peoples, and was the source of the legends of Cyclops, because that narial opening in the forehead was interpreted as an eye. But you can't miss this being an elephant when you look at it. Now, before changing the slide, I want you to realize what it is exactly that you're looking at. This is not an unusually well-preserved specimen, though it gives you certainly the view of the head of one of these animals. This is my digital mastodon. This is a digitized 3D model made from very well-preserved specimens of the skull, the jaw, and the tusks, which are then digitally articulated. And it's going to be an actor in some animations that we'll see later on that will help explain some of the things that I'm trying to talk about. But I just want you to understand that this is the three-dimensional shape of these animals. You will not see very good representations of the three-dimensional shape in most reconstructions, and even in some museum displays. But this is the real stuff. All right. Here are some artist reconstructions of both of the animals we're talking about. On the top, the American mastodon, on the bottom, the woolly mammoth. These, in fact, are just two of the elephant-like animals that lived in North America up until about 10,000 years ago. And they and their relatives lived over many parts of the world, particularly the northern continents, but also some of the southern continents. Basically every place but Antarctica and Australia has been home to elephant-like animals. Some of you may know that the American mastodon was recently declared Michigan state fossil, so there ought to be a little pride and knowledge of that. But it was common, in fact, across most of North America and down into Central America as far as Mexico City and a little bit beyond. The woolly mammoth was a relative latecomer to North America. Mastodons got here about 15 million years ago. Woolly mammoths got here only about 40, 50, 60,000 years ago in the late stages of the Pleistocene. But they were present across much of the northern portion of North America. But there were other mammoths that lived here as well. So we're really just picking here on two of these animals. You're looking now at a 3D model, a physical model of a woolly mammoth. And it's really a very carefully done one. And here you see what the tusks of these animals would look like from, let's say, a hunter's eye, as you're standing in front of this animal. And that is an impressive beast. And what's hard in particular about these tusks for people to represent is they're a complex, not quite logarithmic spiral form that has just a symmetry and regularity which is beautiful to behold. I don't think I'm unique in feeling that way. But is also an interesting and complicated form to represent. But this is a straight up front, first-hand view of what a woolly mammoth would look like. You might say, well, how much imagination is in this? Actually, very little, because this is based on the very well-preserved carcass of the Yukagir mammoth. We'll see him later on, recently found in central Siberia. And we know a lot, in fact, about what these animals look like, even to the point of their body covering. At this site, I have excavated bushels of this coarse hair. Fine, woolly underfur and coarse guard hairs, some of them up to 1 meter long, that represented the insulation, the body covering of this animal. They're really very dramatic beasts. This is from a new edition of Lister and Bond, Mammoths. It's an evolutionary diagram that represents the relationships of these animals. It goes from the past, way up here in the corner, down to the present here along this line. Adrian Lister must have been overcome by wishful thinking, I guess, because for some reason he shows mammoths as extending to the present, whereas mastodons and others of their relatives are shown here as going extinct, as they do just before the present. In any event, in part this is to show that the relatives of these animals, some of the relatives of these animals are sirenians, such as dugongs and manatees and so forth, and the hyrax is even a relatively close relative of them. Of course, the rest of mammalian diversity is less closely related and is not shown here. But the point is that they do have a long history. Mastodons and mammoths actually have a most recent common ancestor some 25, 30 million years ago. Mammoths are much closer, here we are with mammoths, are much closer relatives to the living elephants, the African elephants and the Asian elephant. They are members of the same family, the Elephantidae, whereas mastodons are placed in a different family, the Mammoths, both of them in the mammalian order Proboscidea. At any rate, they have a long history and also a very broad geographic range, as I alluded to before. Can't really go into the details here, but all elephants really originated in Africa and spread subsequently to Eurasia and around via the Bering Land Straits into North America, and some of them even into South America. But many parts of the world had in their faunas as many as 5 sometimes coeval or contemporary elephant-like animals. So what we have today with just 2 genera on 2 different continental masses is really a much reduced, depauperate picture of the diversity of these organisms. Just 10,000 years ago, the world was very different with respect to its inclusion of elephants. Now, how do we find these organisms, their remains in this part of the world? This is, I guess neither of these, as I think of it, is Michigan exactly. This is northern Indiana, this is eastern New York State, but these could as well be sites in Michigan. And those of you that have accompanied me on some of these sites will find this familiar. This is actually the Hyde Park mastodon. It's a view from well above. We had fortunately an arm, a mechanical arm, that sort of cherry picker type thing that extended us out over the site. And you're looking down. Coming right out of the middle of the bone scatter was a spring. And so one of the contingencies of working at this site was to manage the constant flow of water that kept flooding it. And that required a little engineering and so forth. Sometimes the sites are a little drier for scale. Here are my 2 kids, Cara and Noah. They've been helpers in excavating these sites since they were, well, a week or so old, as my wife can vouch. So they're not always quite so white, but it is notable that very often the remains of these animals are occurring in wetland sediments, in clays and peats and so forth that form in what were formerly ponds and bogs and lake margins and things like this. And it's an interesting question just why that is, and I will return to that in just a moment. Once the bones do come back to the museum, there's also an important phase of work that involves just documenting their shapes. In many cases, the arrangements for excavation are collaborative. We don't have permanent access necessarily to all of these occurrences, and so we have to make basically copies of the bones. And there's a complex series of processes associated with molding and casting that are responsible finally for giving us the really complete, well-preserved, nicely mounted skeletons that we are able to show you. There's an enormous number of people that have helped in this. As it says here, 50 plus students, many of them Europe students in the U of M's Undergraduate Research Opportunities Program, many volunteers, and untold thousands of hours to get all this together. But the end product is a really exquisite skeleton of a mastodon that you can see over in the exhibit museum today, well, for the last year or so, and on into the future. This is a really good look. I mean, you can add the soft tissues, I think. A really good look at what one of these animals would be like. Sometimes you see them in reconstructions, sort of looking, their heads down, their tusks sort of barely sticking out, and they look sort of, well, Eeyore-like, if you know what I mean. But this is, I think, a much better picture of what they were really like, with tusks that swept up like this, erect, strong. This is an adult male. These were animals to deal with. This is one finger bone of one mastodon across which you can see some, I'll call them cut marks, but I say that not just because they're straight and long and some sort of incision, but because he looked at them in a scanning electron microscope and seen the sort of substructure that's diagnostic of production by stone tools. Furthermore, the location of these is interpretable in terms of the tissues that cross this part of the bone. We can understand all this in terms of a strategy of meat removal, basically harvesting various tissues from the carcasses of these animals. These animals were important for human subsistence. There is an open issue. I say open, I think there are good answers actually that go each way as to whether these animals were hunted by humans, hunted and killed by humans, or scavenged natural deaths. I think both happened. But I think human hunting was a very important fact of these animals' lives by the time of the late Pleistocene. And as I'll argue also briefly a little bit later, human hunting was probably an important factor in their extinction. But the first step of demonstrating that is just to show that humans had anything to do with them whatsoever. And it's this sort of information, though much, much more than this, that does that. There's this other issue of why the bones of these animals occur in these pond sediments. And old answers to this were as, well, I think goofy as that they were dumb and that they would wander out and get stuck frequently. But again, there's a long argument that would lead up to this, but I think in fact that many of the carcass parts that we're finding were actually put in these pond environments by humans as an explicit planned strategy of meat storage. Now, there's a lot involved with this, a lot of chemistry and biology, and physics for that matter, as to how and why the meat is preserved. I suppose the chemical aspect of it is it's the cold water of the physical is the cold water of the ponds, the chemical, it's the low pH of the ponds, the biological is it's in part colonization by lactobacilli, the same bacteria that we use for making yogurt and cheese and so forth. These combine to actually transform, yes, but preserve the meat and make it accessible long-term for human consumption. In order to test the feasibility of this, two of my good friends and colleagues, Jerry Smith and Catherine Badgley, called me when one of their draft horses died of old age and offered me the privilege of using this horse in an experiment. And what you see here is Jerry and I introducing one of the hind limbs of this horse to a shallow pond on their farm. And we followed this animal and its meat masses over the course of a little over a half year. And some months later, pulling it out through a hole in the ice, even though the meat was looking a little on the side, on the surface, it was wonderful inside and virtually fresh in its condition. But even the next spring, this was in April, it was now growing algae on the surface, but inside, it tasted different, yes, but it was still, I wouldn't say fresh, but it was perfectly edible and a good source of calories for humans. So it's human activity in many cases that was responsible for preserving the remains of these animals. Now, there's another kind of record of these animals' activities and presence in this part of the world that you might not have anticipated would ever be preserved, because aren't footprints on the sands of time they're supposed to be the last thing that they're sort of the archetype of evanescence. But in fact, in certain environments, footprints are preserved. The fossil record has many cases of footprints preserved in it. And this is a case near Saline, not far from here, where we were excavating the carcass of a mastodon, but then on the margins of the excavation began to find these depressions as we carefully peeled down through the sediment layers. And here at an interface between two very different types of sediment, so that we could basically lift one off of the other, was this series of large depressions that we decided in the end could only be footprints. You're looking here down the trackway in the direction of movement of an almost full-grown adult male mastodon. There's interesting features, sort of suction features and so forth, that reflect the interaction of the foot in the substrate. Again, more than I can go into, though we'll come back a little bit to that in a moment. But one of the first questions we had was, now, okay, how exactly is it that a quadruped, a four-legged animal, makes that kind of trackway, which looks, almost just like the tromping of a biped through such sediments? Well, I first have to show you sort of how we documented this in various ways. We mapped it, we photographed it from a bipod, a large set of legs that elevated our camera about 20 feet above the substrate. We documented it most significantly with a fiberglass field mold, which we had to struggle through all vicissitudes of weather and other problems to make. The footprints, when they were being molded, I don't know, there was some deep psychological connection that my son took to these, whether he identified them with Bath or just everyone's center of attention. It's hard to say, but he just loved these footprints and wanted to sit in them all the time. Eventually, we did manage to pick up this fiberglass mold, carry it off to a sheltered place, and proceed to make a cast from it, which is now on display in the exhibit museum. This is the first time in the world that footprints of mastodons had been found. It was not something that we could let go unrecorded and undocumented, so we do have a physical record of it. But the understanding of it, now that's a different matter. In order to understand what we were seeing, what we were looking at, we went down to the Toledo Zoo, where there are some African elephants, through the kind assistance of their elephant keeper, Don Red Fox. We got in with the elephants, raked an area of their pen so that there was a roughened surface, and when they walked over it, they would press out that surface and make very nice footprints, which we recorded in the same way we had the footprints at the site, with our bipods, photographs, mapping, and so forth. And here on the right, you see what's produced in a fast walk, an animal going from bottom to top. This is a legend that shows you that the forefeet are kind of round, the hind feet more oval. And if maybe I could number these, but basically in a fast walk, the hind foot actually passes beyond where the forefoot was. So no feet superimpose on one another. They're all spread out like this, and the pace is extended. But in a slow walk, the hind foot is placed almost exactly where the forefoot was just lifted from. And so you have this superimposition of footprints, and that's what we see at this site. With an exception here, even the slightest turn, the animal here turned to the right and back to the left. Even the slightest turn will bring those prints out of registration. And then you see separate prints. Now, let's go back to our map of that same footprint sequence that we saw at the site. All of a sudden, it looks much more complicated, doesn't it? There's a lot of footprints there. The big ones are the ones that you saw before. Those are deep. But all these others are extremely shallow features. In addition, these two, particularly, and I guess that one as well, of the big prints are very shallow features, totally different from the big deep prints. Let me move on to the next one, which is color-coded, and you begin to see what I'm getting at here. Basically, we've got a number of individuals represented. The big prints are from this adult male, and I use the dark blue for his forefeet, the light blue for his hind feet. But there are probably 3 additional animals, all smaller, all probably adult females, and 1 set of little prints no bigger than about this. That's the purple one, because I don't know whether it's to give it the blue of males or the reddish colors of females. Now, what's going on here? The sediment behaved, here's my physics on the footprints, the sediment behaved as a non-Newtonian material, a material that did not show a linear response to the imposition of stress, that had physical properties that were actually dependent on the rate of imposition of stress. When it was stressed with a single footprint, it would deform in a plastic fashion that yields these very shallow prints. So what about these double prints? The double prints involve the placing of weight by the forefoot, the lifting of weight by the forefoot, the placing of weight by the hindfoot. And that repeated shocking actually fluidized the sediment so that it deformed, making these deep prints. That is actually a very difficult physical system to unravel. What I'm interested in doing is using these footprints, well, in several ways, but right now what I want to talk about is using these footprints as a means of estimating the body weight of these animals. And I can't do that very well with those deep prints. But the shallow prints, now those are a little more tractable. Before we go from this slide, I want you also to think about the implications of the placement of these prints. The big male comes through here, and all the other prints in one place or another deform his. He went through first. The others all came by afterwards, and they all were going approximately in parallel. It seems that they represent a separate group of organisms that probably were going by together. And my argument is that this is evidence of a similar pattern of social organization to what we see in living elephants, where adult males are solitary and females, by and large, associate in matriarchal family groups that collectively raise the calves. And that's exactly what we see here. And these little purple prints are the next generation of mastodons. Now, we want to go back to this issue of body weight. And again, I see I'm fighting the clock, so I'm going to go through this kind of quickly. There's some literature on this particular example of it. Has to do with the interaction between vehicles and substrates, natural substrates. Basically, if you have a four-wheeler, some recreational vehicle like that, and you drive it over the ground, how deep a rut will you leave? Or alternatively, and you realize this is my situation, if you see the rut, can you estimate how heavy the vehicle was? Well, this equation here is a relationship that purports to characterize at least some of the behavior of that system. And to make it short, if you plug in values that relate to our trackway, our animals, our dimensions, our sediment, that's important. You have to characterize the sediment. There are values for these coefficients published for various different kinds of sediments. We get an estimate of body weight that's very nearly 3,000 kilograms. I guess I forgot somehow to take out, one of the other things we did is use a little model of a mastodon. It's behind the scenes, but you've all seen these. And we measured its volume. Well, because we had the skeleton, we could scale it up, figure out basically an estimate of the volume of a live animal. live animal densities, mean densities, yes, bone's a little denser, fat's a little less dense. Mean density is about 1 gram per cubic centimeter. And that actually gave us something pretty close, again in the ballpark of 3,000 kilograms. So I would say that's a reasonable estimate for the body weight of this animal. I was not the first, however, to ask this question. There was other work published that purported to be a solution to what was the weight of mastodons. And the answer given, first of all, the equation here is an equation for mass based on the circumferences of the major limb bones, the humerus and the femur. Now, why should that work? Well, an animal has to support its body weight using its limbs. And so the circumferences of these bones will certainly control their area, and it is their cross-sectional area that provides basically the structure that supports body weight. Those of you that know a little bit more of the physics will notice this exponent, which is 2.5 there. That relates to the fact that body weight in general, as you scale an animal to larger and larger sizes, goes up as a third power of linear dimensions, whereas the cross-section of the bones goes up as the second power of linear dimensions. So you basically have to make bones more robust, relatively speaking, as an animal gets larger in order to support its very much larger weight. Now, the exponent is not 3 exactly. Let's not worry about that. But the point is, this is a nonlinear relationship between bone cross-section and body weight, which should allow you to calculate the body weight of a mastodon. When you run this all through, which we can do, we know our bone dimensions and everything, you get an estimate that mastodons were more like 7,000 kilograms. That's more than twice the amount that we came to before. How is this going to be resolved? The person who did this, Pat Shipman, actually, I forgot to put hers on there, thought that mastodons were actually 2 to 2.5 times heavier than modern elephants of equivalent size, and that affected, she argued that changed their life history variables, that is things like gestation time and time to weaning and so forth. These are things that we're going to come back to. And so the thing we have to judge now is, do we buy that estimate and its consequent effects for these other aspects of life history? Or do we think that our physically based estimate is more realistic? I won't try to answer that quite yet, but I just want to point out that, that there could be some other reason that mastodons had bones that were so robust, that were so overbuilt that we would estimate body weights 2.5 times greater than linear dimensions would otherwise suggest. But keep that in mind. Why should mastodons, particularly male mastodons, be so overbuilt? All right, we're shifting gears. That's the end of footprints. It's the end of physics, I suppose, just for a minute, but come with me to tusks. Tusks are teeth. They're actually second incisors, homologs of our second incisors, but they're incredible teeth. They're teeth that erupt early in life and continue to grow throughout life. They grow by addition of layers of material at their base. I'll show that in a moment. But these layers of material give you, in transverse cross-section, a pattern of concentric rings. I think at least some of you can see these rings here, dark and light layers within the tooth. Certainly here, where the tusk is cut longitudinally, you see a succession of layers. And so these are actually, geometrically speaking, cones of material that nest inside one another in the tusk as a whole. This is a structural diagram of the tusk. So here it is cut down the middle. It has an axis. Here is a conical opening, the pulp cavity, similar to the pulp in your teeth, in the roots of your teeth. And think of the tusk as basically a long, ever-growing root, a root that just grows and grows and grows, displacing the crown outward, way out there in the course of the life of the animal. I haven't put in all the little cones nested within one another, but here's what they look like. And we're now going to look magnified at a transverse cross-section through this. There is a thin layer of cementum on the outside. We're mostly going to ignore that. Dentin on the inside. Here's the pulp cavity. The dentin grows inward as layers are added, cone within cone, extending this tusk in this direction. It erupts out of the socket in this direction, but it actually grows backward. And you'll see that in a moment. What I want to point out here, here's a series of magnified views of the structure of initially 1 year, one of these tree ring-like phenomena in the cross-section of the tusk. There are layers within a year, 52 layers. Anybody ever heard of that number? Within each of these units, of which there are 52, there are more layers, 7. Anybody ever heard that number? And there are 365 of these within what we know to be a year here. Now that's bizarre. We're going to look at a model, an animation of tusk growth in a minute, but I want to show you the last frame of it so that you can be oriented properly. I've forgotten about the cementum. That's this surface layer that we're going to peel off. I'm pretending that the tusk never wears or breaks at its tip, and so you see it all there. But this is the pulp cavity. Each of the color cycles that you see here is intended to represent 1 year in the animal's life, and you see that season follows season, month follows month, and the thing grows in this sort of way. There it is. You're a fly on the forehead of the mammoth, and you're watching the tusk grow under you. It's an invisible mammoth, okay? There you're looking into the pulp cavity, and there it ends. So that's how tusks grow, and I want you to sort of really internalize that when you're thinking about tusks and tusk structure. Now, in this ideal model, of course, every year is like every other year. But in reality, of course, no. Years are different. Parts of years are different. And so we would have to study the whole record in order to get the whole record of life. But the whole record of life is there, except for the possibility of losing some from the tip, which we do. Now, there's another slight problem. How are you going to sample that whole record? Clearly, you cannot do it by just putting one core, say, through the tusk near its base. You have to do one of two things. Either cut the tusk longitudinally and sample it in blocks that you cut out of the material at various positions along the whole sequence, or if you can't cut it longitudinally, you could core it at positions, but you've got to sample it at multiple positions, spaced in such a way that the record overlaps, and you've got to compile in the record from that whole structure. Whoops. We don't really want to see that again. Well, you might, but let's go on. Okay, here is an example of cutting a tusk longitudinally. Somebody asked me beforehand if that was a cast or something I was cutting, because surely I wouldn't cut a real tusk. No, we cut real tusks, stem to stern, 3 meters long and more. It's a great operation, a very difficult operation, but we do it with a rolling scaffold and support structures and so forth that takes hours to days. Here's an example of coring a tusk. This is one of my graduate students, Adam Roundtree, was here this morning, and myself operating the drill in this instance, and we're drilling cores perpendicular to the tusk axis to sample it. This is a thin section, an optical microscopic view of the cross-sectional material of Mastodon ivory. This button is small, and I keep losing trace of where it is. But these are the daily lines. These fine lines that you see here are the individual days. And then the darker bands through here, those are in a mastodon, there are 2-week intervals actually than week. But it's a little more complicated than that. In mastodons, sometimes we see something dark every week, but then there are intervals that are 2 weeks long with not quite. In mammoths, much more consistently, it's a week long for each of these subannual, supra-daily intervals. But they're not crisp. They're a little tricky to see, but that's what they look like. The base of the tusk often shows actually quite clear representation of years. These ridges right here, that's another view of the 2-week intervals in a mastodon and the 14 days in between them. Again, in mammoths, it would be 1-week intervals and 7 days in between them, at least most mammoths. I'll come back to that in a moment. Here you see where each year has been sampled multiple times by drilling a row of holes, actually milling out a groove of material along the growth lines. Each one of those rows of holes then is one sample, and we're interested in measuring its composition. And that composition changes in a regular fashion. Here we're looking at the thickness of these 2-week increments, and each one of these arrows is a winter-spring boundary. Within a year, the thickness goes up in summer and down in fall and winter. There's a lot of noise, there's a lot of variation, but in general, they grow faster in spring and summer and slower toward fall and winter. And underneath each of these years, mapped out by increments, is a year mapped out in oxygen isotope space and oxygen isotope compositions. The two most common isotopes of oxygen, oxygen 16 and oxygen 18, appear in precipitation in different amounts in different seasons. In the summer, we have isotopically heavier precipitation. It's got more oxygen 18 in it. In the winter, we have more oxygen we have less oxygen 18 relative to oxygen 16. And so the value of this oxygen isotope ratio goes up and down with each year. This is a very well-established aspect of modern and ancient climates, and it leaves no question that we are dealing with years in these largest layers of the tusk. All right. I'm 3/4 of the way through my time and halfway through my so this is meant to be a core slide that explains the nature of all of this tusk work. And let me just say that there's a variety of physical observations here, basically structural observations, measurements and counts of these increments, and here compositional observations. It's not just oxygen isotopes, it's carbon isotopes and nitrogen isotopes and strontium isotopes and concentrations of magnesium and strontium and lots of things. But we look at all those, they all vary in intelligible ways because of the animal's diet and behavior and the climate it lives in and everything like that. And so from that, we can deduce things about the animal's biology and environment. Now, we can't always pinpoint everything just exactly, but we can often importantly constrain it so that we know much more about the animal's life than we did when we undertook these studies. You might be surprised at the different aspects of biology that we can get out of this. Just, take for instance, sex. Okay, maybe you can, even though nothing anatomical, soft anatomical is preserved, there are sexual dimorphisms. So that's morphologic, that's maybe not a surprise. But calving, nursing, weaning history, you can really tell how long a mother mastodon nursed her baby? Yes, is a simple answer. What about down here? Well, there's a lot we can learn about the climate from studying this. It's not a mystery. It's basically that the tusks are made of materials that are consumed by the animal. What comes in, what goes out, leads to these fluxes of materials that change with the seasons and times and conditions. And that leads then to an understandable sequence of layering in this wonderful archive of life history that is the tusk. And that's our animal again. So it holds out on its face there its life. It's basically its whole life for us to study. Now, this animal, this particular bushing mastodon, if you look, I guess you can't see or maybe you could see my arrow, but here I'll do it with a laser pointer. Right up here is a hole in his skull. There it is, a close-up. This is his right wisdom tooth, upper wisdom tooth. That is not normal anatomy. That is a round hole punched through the bone. Well, he's not the only one that has that. And it led to the idea that, like living elephants, male elephants in must, this unusual hormonally mediated sort of rutting season they get into. I can't in Miss Company even tell you all the things about elephant must. But one of the things is that they get very aggressive. If they encounter a similar sized male, they'll basically say, "I want her." And the other one will say, "No, I want her." And they'll go at it. And the dust you see rising from their backs here is the shock of impact as these two ram together. And they will sometimes kill one another. But that's elephants. Did mastodons do the same thing? Well, let's see. First of all, is it possible, given the different tusk geometry of mastodons, to even sort of figure out how the tusks did something like this? And remember, I've got not just this one, but about a half a dozen of these animals that have holes in the sides of their heads. This is a single frame from an animation that we'll show in just a moment, showing just the moment before the strike. The one on the left is going to puncture this region of the tusk from the one on the right. And yes, I think it's clear they could do it. We used these very accurate 3D models to actually, I mean, it's maybe not that different from what you did when you were a kid with your little toys, you know. But here they are, a little premonitory fencing, get into a sort of wrestling match where you try to twist the other one so it's off balance, off guard, and then raise that tusk tip up into the cheek of the other one. And that's basically, if not a fatal blow, and it sometimes wasn't, it's a very damaging blow to, if nothing else, the masticatory musculature. You're sort of out of commission on that side of your mouth for a long, long time. This is actually a close-up view of the base of this tusk, which is one of these animals that met this sort of end. And as I was looking at this cast, I had noticed these features on the original specimen, but the cast almost made it more clear by factoring out the color variation. There's rows of pits here that are not all the same length. This is a short one, so that I don't have to stand here and convince you that. I'm just going to put up red lines that represent the positions of these rows of pits. On the flank, the outside curve, the basal region of the tusk here, they're centered on about the same line following the curve of the tusk around. But they are different lengths. We know it's a little puzzling because the cementum, all of this surface here, is accreting layers of material. How is it that those pits have stayed there? They follow the growth lines. They clearly, I think, represent something that was done when this was the growing margin of the tusk, which it now is not. So the tusk has grown beyond where they are, and yet they remain at the surface, even though the surface at the time they were made is buried deep within that. It's a bizarre sort of series of problems. Just so you understand the seasonal significance of the topography here on the tusk, there are letters spring, summer, fall, winter, spring, summer, fall, winter that show how seasons relate to that tusk topography. Each one of these arcs of cementum defects, as I'm calling them, is located at the end of spring, the beginning of winter, the same time of year. Each one represents a little place where something has happened in tusk formation. Do a thin section of that, look at it under the microscope, and this is the three-dimensional characterization of that cementum defect. Basically, the pattern of lamination of cementum in this area is disturbed from the beginning of cementum deposition, and it never sort of calms down. There's likewise anomalous dentin that's formed in this from the beginning of dentin deposition. So what's happening? Well, this is a more close-up view under one of those areas of defects. There's a little, this is the growing end, this is the tip. There's a little piece of material bent downward. And so I'm going to back up time and remove the cementum above this line and the dentin below that formed subsequent to the time when that was the growing margin of the tusk. Here's the growing margin of the tusk in the base of its socket. I know this is the outside and now it's up, so it should be down, but you get the picture. What's happening, I think, is the tusk base is being driven into this bone at the back of the socket, like driving your car too far into the garage. And what happens? Bumper damage. Well, the growing margin of the tusk is actually physically damaged, and some of the cells in that position then are injured in a way that they never after produce normal tissue. So the abnormal tissue is a record of that damage that actually was sustained at the time that was the growing margin of the tusk. Now what we have here is our model chopped in a plane so that you see it's down the plane of the tusk socket. It's going to rotate around and show us a little better what we're looking for. So here it rotates, you see the plane of section, and now, gunk, gunk, gunk. That's the tusk actually displacing, I've exaggerated it of course, displacing due to the reactive force acting on the tusk tip that arises because he's just jammed his tusk up into an opponent. You jam your tusk up in, there's a reaction force that goes down and actually displaces the tusk in this direction. It rotates effectively by a point up here, because that's where it's best supported. But what the consequence is that this part of the growing margin on the outside curve of the tusk is levered into the back of the socket and produces that damage. That damage is then a record of battles. It's a record of battles throughout this animal's life. And whoops, again, we don't want to do it again. Okay, here's a series of samples of that animal's tusk. There's about 34 years. I just looked at the clock. A lot of work goes into piecing together this record, but let me shift here to, this is to be read like this is a graph that's long, 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 because it's all the years of the animal's life arranged as music here. These are the ups and downs, the weekly growth rates of that animal's tusk, and there's a lot to be seen here. This, can you see that? Yeah, that is the year of his maturation, when he was evicted from the matriarchal family unit. It was a hard time for him, and the tusk grew only very little. There were areas here, now shown in red, those were where the marks of the fight were. And that represents the onset of muss. So we see here good physical evidence of events in his maturation and life history. And this is one frame of another animation that's not finished yet, but it's another dramatic look at these animals in the course of a fight. I want to, we're going to have to cut down on mammoth material, but before we leave mastodons, I do want to say something about extinction. It's not that only mastodons went extinct, no, mammoths did too, but our evidence right now for mastodon extinction is better. We've got more of it than we do for mammoths. And this is not all of it by any means, but let me just show you here with data from living African elephants that calving interval and age of maturation are sensitive indicators of the kind of environment the animals live in. Under good environmental conditions, they mature early and they have short calving intervals. Under stressful environments, they mature significantly later, almost often the age of 20, and have calves that are spaced out much more in time. So there's an interesting question. What were things like for mastodons and mammoths at the end of the Pleistocene? And I'll just tell you now that the preliminary data that we have is that the animals matured around 10 and had calves spaced at intervals about 4 years. Sounds like a good environment to me. One of the explanations for extinction, however, is that there was some sort of deterioration of their effective environment. And I would argue that that's not consistent with the evidence of age of maturation and calving intervals that we see. Here now is data on, or are data on mastodon and just a couple mammoths, age of maturation at different points in the past. These are radiocarbon years before present, 12,000 years ago, 11,000, 10,000. This is about the time of extinction. Age of maturation is going down as we approach the time of extinction. That's just the opposite of what we would expect if it was environmental deterioration causing the extinction, but it's exactly what we predict if there was an increase in mortality due to hunting. There's data on elephant seals that represents one of many modern analogs of this. The bottom line is that the patterns of life history are changing in a way that supports hunting as the cause of extinction, not climate change. Before we end, I do want to spend some minutes talking about Siberia. I went there initially almost only because there were such well-preserved specimens. This shows something of a landscape. It's the most incredible country you've ever seen. I mean, the scale of it, the beauty, it's different than anything you've ever seen, but it's really quite amazing. Its mammoths are wonderful. This is at the Yucca Gear site. Here's the sole of the foot of a mammoth. This part of the leg is the meat, it's red meat, it's preserved just incredibly well. The head has been sort of compressed and mushed a bit by freezing and thawing and so forth, but those are its soft tissues. That's its ear, its eye, its musk gland. He's all there, basically. Not all the body, but all the head. These are some of the tusks that had been recovered by some of my colleagues. It's a wonderful place to go to study these archives of life history and so forth that are retrievable from the tusks. Some years back, this was all stored in pretty primitive circumstances. They've now really gussied this up a bit. It's impressive, but I don't have time to tell you everything about it. I better move on. This is my lab just outside the door of that ice cave. Yeah, okay, it's under the city dump. It doesn't look too great from this side, but look at the view. This is looking north out across the river and over the tundra, and I tell you, many fine hours of drilling and thinking about mammoths have been spent here. And what do we see when we see a tusk in cross-section? We don't usually cut them, but here one was broken, and we were able to look at those annual rings, just as clear as you could ever ask for. There's a really interesting pattern, a departure from the ordinary that I've described to you so far. This is temperate latitude mammoths, always these weeks, always 7 days, always 52 of these weekly lines per year. But in the Arctic, in some of the years of the tusk, particularly the last year of the tusk, springs and autumns look like lower latitude configurations, but summers and winters have what looks like 11 days in the week, and they have fewer weeks, if that's still what they are, in a year. What does that mean? Here actually are, I guess you can see that, maybe this is quicker. Here is the 7-day pattern with 7 daily lines per each one of these weekly lines. Here is the 11-day pattern with 11, you could count them per each of these weekly patterns. What's going on? Well, I think it has to do, I would argue, with beat frequencies. And here we come back to a little bit of physics, and maybe there's just time for me to switch to the oscilloscope and for me to sound these wonderful tuning forks that the demo lab provided. Here is one clear tone of one tuning fork. Now, these are actually manufactured to be essentially the same, but a little bit of clay has been put on one so that it damps its vibration. But it's a clear tone, no modulation of the amplitude of any of that. Perfectly clear tone. Other one, also a clear tone. For all intents and purposes, it sounds the same, right? But in fact, the clay makes one of these vibrate at a slightly different frequency than the other. And you can hear that because they constructively and destructively interfere in quick cycling when they both are sounded together. You hear that? And the oscilloscope is showing it to you. And I can actually change it to yet a different value now by adding another little piece of clay. Clear. Different frequency. There it goes, different frequency, because now there's a different beat frequency. The beat frequency is the difference between the two fundamental frequencies, and it reflects the changing relationship between those two fundamental frequencies. Now, what frequencies are we talking about for mammoths? Well, one of them is, I think, a circadian rhythm, which is under ordinary circumstances, we think, a 24-hour rhythm. Okay. But here's the kicker. It's a 24-hour rhythm only when there are external stimuli to entrain to that recur on a 24-hour cycle. And at high latitudes in the winter and in the summer when there's no night or no day, whichever, you don't have that environmental cue. What happens then? This is a human in a cave with no external stimuli. If the human, and these are the activity patterns, sleep and awake for this person. If they went on a 24-hour cycle down in the cave, their activity pattern would run straight down this graph. It shifts because they actually express a longer rhythm. That is the endogenous or internally controlled native rhythm, if you will, of their circadian system. So it's expressing its own rhythm without the environmental stimuli to entrain to. What I think is happening, in short, is that under winter and summer conditions for high-altitude mammoths, they go into this free-running phase of operation rather than an entrained phase. There's a bunch of math here. I'm just going to have to go through it. This is just to say that given this configuration and the light regimes at different latitudes, the farther north you are, the more of the year you're in the 11-day mode rather than the 7-day mode. And so there's a consequence that actually is plotted out here. there's a dependency of the number of beats or weeks, these physiological weeks per year, on where you are in latitude. And without taking, I really do have to click by some of these, but let me just go to this one. This is actually two tusks, two individuals. They're two males. I've registered them or overlapped them at the point of maturation here. And it just shows you that within a single tusk, you actually vary from conditions typical of the Arctic to conditions typical of temperate latitudes. That means movement. That means these animals are going north and south. And in the years before maturation, they're going north and south more frequently on about, it turns out, a 4 to 6, or maybe in the worst case, a 7-year cycle. And then as an adult, as a male adult, it goes north and south. Here it's going north, there it's going north for the first time in its mature life, less frequently. Basically, the north-south movements for females are on a calving cycle. They're going north to have calves, and it's very regular. And even the males go north and south with their mothers when they're immature. When they're mature, they go south to grow up, get strong, get ready to go into muss, and then they go north only when they're going to go into muss. And they exhaust themselves, come south, and recoup until they go north again. So there's a lot of interesting life history that's encoded in the tusk from one end to the other. Okay. I'm getting near the end here. To wrap things up, it's not only adults. We're also interested, of course, everyone's interested in the younger generation, in the calves. But we have an extra interest in the calves because remember, sometimes there is loss of material from tusk tips. And we want to have a way of estimating what that is. What we need really is some marker that has some well-known or at least knowable relationship to the beginning of life that's near the tusk tip that we might still see in the tusk of an adult. Well, we have a couple options, but really one of them is going to come through for us. Let me just tell you and keep you in suspense since time is short. The marker is weaning. Weaning occurs, maybe it's a variable age, but it's a relatively well-constrained age. And if we can find a weaning signal in the tip of a tusk of an adult, we know basically how many years are lost from the tip. This is a wonderful little calf. Unfortunately, it was chopped in about a third, and only a third recovered. It was found in a gold mining operation in eastern Siberia in 2004, the Oymyakon baby. And knocked loose from its face was its premaxilla, the part of its snout that embeds the tusk, and there, with a piece of bone removed, is the tusk. And here it is pulled out. We're going to zoom in and look right here within this rectangle, and there's what it looks like. Here toward the tip is light-colored material that represents dentin that was formed before a certain point in time. This line is a sort of time marker. Here's darker dentin formed after that. This line, we think, is what's called the neonatal line. It marks the sort of physiological stress of birth. And the part to the right was formed before birth, the part to the left formed after birth. This is a longitudinal section through that tusk, and right here is a well-marked growth line in the cross-section of the tusk that corresponds exactly to that color change in the exterior. That represents the time of birth of the animal. Toward the tip is the material formed in utero. Toward the base is the material formed in the early years. Well, this one only lived 8 months. I slipped there and said he, but I'll let you in on something. We've done DNA work. We know it was a male. There's a lot that we can tell from these animals, and again, it's a whole other talk to talk about. So, this is Lyuba, means love in Russian. We estimate about a 4-month-old female mammoth calf, and she is the most perfectly preserved mammoth ever. Her earlobe has been gnawed off, probably by Arctic foxes. Her tail has been gnawed off by Arctic foxes, and that's all that's wrong with her. Here she is. We're treating the surface of her hide with a formalin solution to just suppress any current biological activity going on. She's being maintained in a frozen condition so that we can do all the DNA studies and histologic studies and all the other studies that we want to do. And I leave you then with just an acknowledgement of the many people that have helped in this, both here at the University of Michigan and colleagues elsewhere, and that's a good place to stop. Thank you.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.